Welcome back, Form and Function students. We're here again for another video lecture on a snowy day. Today we're going to continue our discussion of internal transport, and this is the last lecture in that section. So we're going to begin by talking about blood and various aspects of blood and its importance and relevance to internal transport in animal systems. So to place it in context, so internal transport systems between animals and plants is very similar. So they're transporting some of the same materials throughout the systems. The only major difference being that in animal systems, the internal transport system is going to be transporting heat throughout the body, whereas plants clearly don't do that. But they do, in both systems, transport gases such as oxygen and carbon dioxide, being the most important. They're going to transport nutrients throughout both of the systems. They're going to transport waste products so that they can be removed from the systems. And in both, they're going to transport hormones throughout and send them to targeted areas where they'll be utilized, primarily as signaling devices. And then they're also involved in the immune system. Now, in the case of blood in animal systems, and right now we're primarily focusing on the vertebrate system, blood is composed of an extracellular matrix referred to as plasma, which makes up about 55% of the content of what's referred to as blood. And then there are several formed elements within blood. So those formed elements consist of platelets. You can see these here in these light-colored cells here on this slide. So platelets are these cells that are important in the immune system. We're going to talk about it more when we get to defense. But the short version of this is that they're very important in clotting. So whenever you have an injury, these are cells that rush to the scene and then begin to form a clot to prevent further blood loss from happening. White blood cells, which you see indicated in the slide here by these different purple cells, are part of the immune system, and we'll talk about it more when we get to defense. And then we have red blood cells. So red blood cells transport oxygen and carbon dioxide in animal systems, and they do this primarily via a protein called hemoglobin, which has some really interesting properties in animal systems, particularly in mammal systems. And we're going to go into much more detail about hemoglobin in this lecture. By the way, it's important to note that in mammal systems, red blood cells lack a nucleus, but in virtually all other animal systems, they do possess a nucleus. So let's talk about hemoglobin. So you can see a picture of it indicated here. So it consists of 4 polypeptide chains. So it has 2 alpha chains and 2 beta chains. And each of these chains contains a non-protein group called heme. And that's indicated here by this sort of yellowish rectangle. So each heme contains an iron ion that can bind an oxygen molecule. So in the case of this hemoglobin molecule, it can bind up to 4 oxygen molecules, 1 on each of these heme groups. So when you have 100% saturation of hemoglobin, that means that every possible oxygen has been bound to each of the 4 heme groups. So in blood, within the red blood cells, 98.5% of the oxygen is bound to the hemoglobin. So approximately 1.5% is dissolved in the plasma. Now, as it happens, oxygen doesn't dissolve very well into water. And so if your body was dependent upon dissolved oxygen in your, say, plasma to be transported to where it needs to go in the body, it would be incredibly inefficient. You couldn't get enough oxygen into your plasma to work. So hemoglobin is an amazing molecule that has great properties for carrying around a large amount of oxygen. And it's also important to know that in your body, so red blood cells are produced in your bone marrow, and you humans produce approximately 2.5 million red blood cells every second, and approximately the same amount are dying every second, which makes sense, right? So hopefully you've got a balance there in the number that are dying and the number that are being produced. And new blood cells will live for approximately 120 days. So they can live for a while, but you are constantly producing new red blood cells all the time. And that's great because you're going to need all those red blood cells to carry around hemoglobin to power all of your different organs and muscles. So in this diagram, they're showing you a schematic of oxygen going from the lung, where it could be 100% saturated, moving through just those few cell layers, and then being loaded into red blood cells. And then those red blood cells are going to move throughout the body, and it's going to offload that oxygen wherever it's needed in the tissues. Now, the rate of that unloading is going to depend on the partial pressure of oxygen in the tissues. So generally speaking, the partial pressure of blood that's in the lungs or that's leaving the lungs is 100 milligrams of mercury. In tissues at rest, it's generally hovering at 40 millimeters mercury. So that automatically is going to be setting up a diffusion gradient. And as you already know, and we visited several times, that means oxygen is going to move from an area of higher partial pressure to an area of lower partial pressure and then bring that level up, let's say in the tissues. But let's look at how that works in a little bit more detail. So this is a diagram from your book, and what it's showing you on the y-axis is the percent saturation of hemoglobin running from 0 to 100%. And on the bottom, on the x-axis, is the partial pressure of oxygen in the blood within tissues. So again, going from 0 to 100%. And what should immediately strike you is that this the representation here is a sigmoidal curve. It's not linear. It's a sigmoidal curve. And notice that when the partial pressure of oxygen in tissues is low, the percent saturation in blood is low. And when the partial pressure of oxygen in blood is high, the percent saturation in hemoglobin is high. And there's this very, very steep curve. So initially, this curve strikes many people as being non-intuitive. Well, you would think that, so remember what we just said. So at rest, generally, oxygen in tissues is hovering at about 40 millimeters. And you would think that if tissues are going to be low, you would need to have a situation where the saturation of hemoglobin is high. But it actually doesn't work that way. Let me explain to you what's happening here in all the different parts of this curve. But it's important to note this right here. So where the steep curve is, a small change in the partial pressure leads to a large change in how much oxygen unloads from hemoglobin. So hemoglobin has this really interesting property of being able to load oxygen quickly and unload it quickly. And that property is referred to as cooperative binding. So remember, you have these 4 heme groups, each of which can carry an oxygen molecule. So cooperative binding is a phenomenon where once you have one oxygen molecule that's become bound to a heme group, that makes it more likely that other oxygen molecules will bind as well. And in the reverse fashion, once one oxygen molecule is released from hemoglobin, that makes it more likely that other molecules are going to become released as well. You can think of it as being kind of like a party situation. So maybe someone that you really don't know very well is having a party. They're planning to have a big party. And at first, maybe no one's really there. The party begins. But a few people, because they got nothing really better to do, a few people decide to go. And they go and they realize it's a great party. Like everything you could possibly want is there. They got food, they got drinks, they got fun activities happening, they got a great band going. And they start tweeting out that, hey, this party is rocking. And as soon as that message goes out and a few people go, then more people go, and then a whole bunch of people go. And the more people that go, the more other people will go. And suddenly you've got a rockin' party going on. And then by the same token, at maybe dinner parties or even big parties, once you get to a certain part of the night, once a few people start to go, it's like all of a sudden everybody takes that as their cue to leave the party and go home or go someplace else. So that's an analogy for what's happening with cooperative binding. So if you look at this figure then, remember we said that 40 is about the partial pressure of oxygen in tissues that are at rest. When you start and if you trace this figure upwards and reach the sigmoidal curve and trace it over to see what the saturation level of oxygen hemoglobin is, it's about 75%. So what that means is there's a huge amount of saturation here that will unload oxygen into the blood and keep it oxygenated. When you're in deficit, so when your muscles are exercising, say you're at the gym, then that means so oxygen is being used up, you're in oxygen depletion, and that means your partial pressure in tissues is going to be lower. So it's going to be in the 30 or 20%, okay? So if you look at this curve, what this is showing is that this is showing exactly what cooperative binding is. So once you start binding oxygen, more and more oxygen is going to quickly be bound to the hemoglobin. And once you're in a deficit situation, more and more of that oxygen is going to be unloaded from the hemoglobin and go into your tissues. Let me show you another diagram that'll probably explain this a little bit better. So here you are with that cooperative binding curve. And here are the lines now drawn for when you're exercising. Let's say your tissues are at 30, partial pressure of 30. And here they are at rest when they're at 40. So when your muscles are exercising, if you trace this diagram up, so you go up when you're resting, and here you are at about 76, 78%. So if your muscles are resting, then your blood is actually depositing about 25% of its oxygen into these resting muscles and fully oxygenating them. When you're exercising, so trace this line up, so now you're at about, call it 58%, okay? And your muscles could be exercising right up to this point as well. So now, if you're at 58%, then that means about 42% of the oxygen that would be in the blood is offloading its oxygen into the tissues, okay? And so what you notice is that there's a big difference in the amount of oxygen that's being offloaded into resting tissues versus what's being offloaded into exercising tissues. So there's a big difference between here and here. And so this sort of highlights what cooperative binding can do. So when tissues are exercising and they're in oxygen deficit, hemoglobin will unload its oxygen really, really quickly and dump it all into the tissues to bring it where it needs to be. And then once it's where it needs to be at 40 or above, then oxygen will begin rebinding to hemoglobin and will bind quickly. Compare this, by the way, to a situation where you have non-cooperative binding. So notice that there's a much smaller difference in the amount of oxygen being offloaded at exercising versus resting tissues. It's about the same. And so this is no good. If your tissues are in distress, you need oxygen to get into them very, very quickly. And so that's why this curve is incredibly steep. So it's trying to get oxygen to your tissues, and it is getting oxygen to your tissues really, really quickly, and then plateauing off. Now, there's another interesting phenomenon that occurs with hemoglobin, and that's called the Bohr shift. So when you have any kind of changes in pH, so a decrease where your tissues become acidified, or if you have changes in temperature, so you have an increase in temperature, that will shift the sigmoidal curve to the right, which means it's going to be even more likely for oxygen to be released into your tissues at all levels. So regardless of where you are in this curve, you're going to be more likely to release oxygen into your tissues at all values. So that's great, because when you're exercising, your tissues, of course, will produce an enormous amount of carbon dioxide, and I'll explain part of that in just a minute. That will cause the pH to go down. And then when that happens, your tissues are in desperate need of oxygen. And so the Bohr shift shows that your hemoglobin will then offload oxygen very, very rapidly into those tissues to allow those tissues to recover. Another point to note, by the way, is that fetal hemoglobin has a higher affinity for oxygen than adult hemoglobin. So oxygen, as a result, will be transferred from the mother's blood to the fetus's blood to accommodate for that need for oxygen. It's not surprising, given that with the fetus is very active, it's growing, it's going to need a lot of oxygen. Okay, so let's talk about that process in even more detail. So remember that red blood cells are not merely transporting oxygen, they're transporting carbon dioxide as well. And so carbon dioxide is going to be released from your tissues as a function of that respiration process. So when carbon dioxide is released from the tissues, it's going to go across these cell membranes, and it'll enter into red blood cells. But it's going to get transformed in those red blood cells. So red blood cells aren't carrying around carbon dioxide itself. Carbonic anhydrase is going to change. It's going to transform carbon dioxide into bicarbonate ions. When that happens, bicarbonate ions are going to be transported out of the cells and into the plasma, and the plasma is what's going to carry those bicarbonate ions to areas where they're going to be released. And I'll explain that in a second. At the same time as those bicarbonate ions are generated, there's also going to be a large number of protons that are produced, and those protons are going to be bound to hemoglobin, which actually facilitates more oxygen to be absorbed by red blood cells. An interesting phenomenon too that's happening, so as this chemical process is taking place and bicarbonate ions are being produced, I can show you on the next slide. So when the partial pressure of carbon dioxide in blood drops, so remember, carbon dioxide is being transported into the cell. When it's in the cell, carbonic anhydrase is going to create, ultimately it's going to create bicarbonate ions. Those ions are going to get transported out of the cell. So as that process is happening, that means the partial pressure of carbon dioxide is going to drop in the blood, right? So it's being converted and it's being exported out. When that happens, that creates a strong partial pressure gradient between the inside of the cell and the outside of the cell, which is going to favor carbon dioxide being transported into red blood cells to be converted. That's actually a really good thing. So yeah, this is what that's showing. So you're going to have more bicarbonate ion being generated. That's great. And then as a result of that, what's going to happen is there's going to be, because of those protons, right, there's going to be a decrease in pH. It's going to kick off a Bohr shift, which is going to make it much more likely for hemoglobin to release oxygen into the system, right, because of this diffusion gradient. What then happens is a signal goes to the brain, which sends a signal to your body, to the respiratory system, to start increasing respiration rate, because it senses that there's an increase in carbon dioxide and you need to release that carbon dioxide, right? So these protons are produced that are going to initiate that increase or that shift in that Bohr shift. And then what happens is in the alveoli of your lungs, a partial pressure gradient favors the diffusion of CO2 from the blood plasma and red blood cells, but that's as bicarbonate ion. But it's going to cause a partial pressure gradient that's going to favor carbon dioxide leaving those cells going into the alveoli, and that increase in respiration is going to cause you to expel that carbon dioxide into the environment. When that happens, that's going to increase this drop in blood partial pressure carbon dioxide, and that's going to cause a reversal in the chemical reaction that occurred in the tissues. So then you'll have bicarbonate ions that are then transformed back into carbon dioxide and water, right? And then that's how it's going to get released through your lungs and out your mouth. It's an amazing process, right? This is killer how this works in the body. Okay, so what this means then is that hemoglobin molecules act as a buffer and helps to minimize changes in pH in their body. So remember, this is all about homeostasis, right? What happens when your body isn't at all of its ideal set points? Remember that sensor, integrator, and effectors. So when the body senses changes in pH or in partial pressure gradients or in temperature, the body is then going to accommodate that, right? So going back here, these protons are going to be produced, bind to hemoglobin, cause a release of oxygen, cause changes in your respiratory rate, so on and so forth. Okay. So now let's ratchet out a little bit and talk about closed circulatory system functions and how they supply tissues with oxygen and nutrients and remove waste. So we've already talked about this a little bit. We've talked about open circulatory systems, and in lab this week you would have been talking about closed circulatory systems, and so maybe some of you will get to go to lab tomorrow. We'll see what happens. But in this stylized diagram, right, so you know that blood is flowing through this continuous circuit of closed vessels. So here's a diagram of a two-chambered heart. You're going to have blood that then flows out, usually to what's referred to as a pulmonary circuit, where it's going to get oxygenated, and that's why it shows up as red traditionally. It doesn't actually turn red or blue in your body. That's just how we indicate oxygenated versus blue deoxygenated blood. So it goes to some sort of pulmonary circuit, picks up oxygen, goes out to what's being stylized, being showed here diagrammatically as being the body. The body is using it, and it's going to use all those gases, and so it becomes deoxygenated, travels back to the heart, and it continues. So you have this continuous loop. And within this system, pressure is provided by the pumping action of the heart. So you need some sort of pump that's going to keep this fluid flowing around. In a closed system, you also frequently have, in an open system, you have a heart pumping as well, but it's just sloshing that hemocyanin or whatever the circulatory fluid would be. It's just sloshing it around the organs in the body of, say, like insects and flatworms and various invertebrates. And it's a rather inefficient system because it's not consistently taking it to a place to get oxygenated and then taking it out to where it needs to be used. So you have a mixing of oxygenated and deoxygenated blood, which isn't very efficient. So in this way, blood flow can be directed in a precise way to respond to tissues and the body's needs. So we're focusing on vertebrates, but truth be told, closed circulatory systems can be found in vertebrates, in annelids, so hopefully you remember that those are various kinds of worms like lumbricus, so earthworms and others, but you also have leeches in that same system. And also in cephalopods, so squids, octopi, cuttlefish, various of those sort of upper level mollusks. And so here's sort of a diagram showing you stylistically the way the circulatory system works in, say, an octopus, which has branchial hearts, but it's still a closed loop system. And then you're seeing some of the auxiliary hearts that are viewed in earthworms. And then here's a stylized diagram showing you the loop system. Now that said, there's a variety of different heart structures in closed loop systems, so closed circulatory systems, and we'll talk about those in just a second. So of course, the closed system is composed of arteries, veins, and capillary beds. In the case of arteries, in this stylized diagram, what you see is, generally speaking, arteries are thick, much thicker vessels. They're tough, and they're going to take blood away from the heart and towards the either pulmonary circuit or systemic circuit. Pardon me. Okay. Now, you can get variously sized arteries, and smaller arteries are referred to as arterioles. And you can see the components, the different layers of arteries. So you have fibrous tissue on the outside, you have muscle tissue, you have elastic tissue, and then inside it's going to be lined by the endothelium. Now, arteries need to be thick and tough because, so it's going to be taking blood from the heart, which is going to be pumping at a pretty intense pressure. And so these are vessels that need to be able to take that pressure and not burst or be damaged by the blood flowing at high pressure through them. So I say that here. So the walls of the arteries are composed of smooth muscle fibers and elastic fibers. So it is able to stretch a fair bit, but they're tough. And so it can expand under high pressure, but it won't burst. So the muscles in these vessels control the diameter. So in physics, that's not hard to understand. When the muscles contract, the diameter is going to decrease, but that's going to cause the pressure inside that artery to increase, right? So it's going to increase pressure. And when the muscles relax, the diameter is going to increase. Blood pressure is then going to decrease. So generally speaking, arteries are going to carry that blood away from the heart to some area, either to get to pick up oxygen in a pulmonary circuit or in the systemic circuit to your organs. And when it reaches both of those sites, those vessels are going to get smaller and smaller until you reach the smallest type of vessel, which are capillaries. So in the case of capillaries, the walls of the capillary are going to be about one cell layer thick. So it's going to make it very easy for gas exchange to occur, either to pick up gases and or to release gases, and to also release nutrients to where they need to go. Then, of course, once they've served their purpose, blood is then going to be carried back from veins. Veins carry blood back to the heart. They have a tendency to be much thinner than arteries. And generally speaking, blood pressure is going to be lower in veins than it is in arteries. Now, small veins are called venules, and it'll get a little bit thicker as you move towards the heart, but not that much thicker. So what I'm indicating here is all the same sorts of layers found in arteries, you'll also find in veins, with the exception of elastic tissue. So these are vessels that aren't as elastic or as tough as arteries. So they have thinner walls and larger diameters than arteries. Once again, blood is under lower pressure. And in the case of veins, so with arteries, the pressure coming from the heart is intense enough to keep that blood flowing in that one-way direction where it needs to go. But once the blood has gone through the capillary beds and is coming back towards the heart, blood pressure is low enough that you have a risk of there being backflow. So as the heart is pumping, blood can be going through the veins, backing up. So to prevent that from happening, you have valves. You have little flaps of tissue in your veins that help to prevent blood from backflowing as it's moving towards the heart. Now, it's also true, and we're showing a diagram of the lower leg here, but skeletal muscles, as you're using them, as you're moving around, they're going to compress large veins, and they're actually going to help blood flow through the body. So what they're showing you here is a large calf muscle, and you have these deep leg veins. As the muscles contract, then that helps to actually push blood through these vessels. And you can actually see the flaps here too. So here's blood flowing through, in this case, in part from the pressure of the calf muscles. And then when the muscles relax, you can see that these valves then come down, and it prevents backflow of blood backwards through the veins. So we're gonna skip talking about the lymphatic system, so you can cut this out of your reading guide, this information. So it's numbers 21 and 22. Okay, so now let's explain how the heart works, and we're going to talk about various different forms of heart structure in vertebrates. So in animals with closed circulatory systems, the heart contains at least two chambers. So you have an atrium, which is going to receive the blood that's returning from the body or the pulmonary circuit. And then you're going to have a ventricle, which is going to push blood out into the system. So generally speaking, the atrium is going to be smaller and have thinner walls. It's merely accepting the blood that's returning to the heart, and it's going to push blood into the ventricle. Generally speaking, the ventricle is going to be much thicker, and this is an intense muscle that's going to need to have a pretty substantial force to eject that blood into the system and keep it moving around the entire body. Now, the atrium and the ventricle are separated by valves. You can see them right here, referred to as the atrioventricular valves, which makes sense. Now, in fish, there's a single circuit, and this is a fish circuit that you're seeing right here, kind of stylized. It's generally a two-chambered heart, so a single atrium and a single ventricle. And so blood is going to be oxygenated on the way to the body. So blood will flow from the atrium to the ventricle, out to the gills to pick up oxygen, and then it'll pass through a capillary bed in those gills, then go into the body, and it's going to transport to the body, and then from the body it'll go directly back to the heart and continue. Now, in the case of, say, frogs and lizards and a few other organisms, there can be two circuits. You can have a pulmonary circuit that goes to the lungs. So it'll go from the heart to the lungs. It will then come back to the heart and then get pumped out to the systemic circuit or the body. So let's keep it straight, by the way. Arteries go away from the heart, veins come back. So you're going to have an artery that's going to go out to the lungs. You're going to have a vein that comes back to the heart. You're going to have an artery that goes out to the body and veins that come back to the heart. Okay? Don't get confused about the fact that this is red. So some people think arteries red, veins blue, arteries oxygenated, veins deoxygenated, right? But bear in mind that this is going to be a pulmonary artery going away to the lungs. Pulmonary vein, so this is a vein that's bringing blood back to the heart, but it is oxygenated. And then you're going to have a systemic artery that's going to go out, usually the aorta, and then you're going to have a vein bringing back. Just want to make that clear. So in this case, you have a three-chambered heart. So amphibians, turtles, snakes, and lizards have a three-chambered heart. And so what's happening here is you have two arteries. You have a single ventricle, and it doesn't have a septum in it that's separating out deoxygenated blood from oxygenated blood. So what that means is, so blood is going to flow into the, and we always refer to anatomical orientation. So blood will flow into the right atrium, and then in this case, for these guys, for these three-chambered, it just goes into a single ventricle, and then it goes out to the lungs, comes back to the heart and goes into the left atrium. And I know on your screen it looks like right, but remember we're thinking anatomical left. So it goes into the left atrium, back into the ventricle, and then gets pumped out to the body. So what will happen is you will get some mixing of oxygenated and deoxygenated blood in the ventricle. What this diagram isn't showing you, but we're going to explain if you take my herpetology class in the fall. There actually are mechanisms. There are structures in the ventricle that helps to direct more of the oxygenated blood out towards the aorta. And then in the case of the pulmonary circuit, there are structures that try to focus the deoxygenated blood going out through the pulmonary artery. But we'll talk about that in class this fall. But the benefit, as you can see in this case, is that you have a separate oxygenated, pulmonary circuit that efficiently picks up oxygenated blood and helps to at least partially separate that and then send that oxygenated blood directly to the body. And then, of course, in birds and mammals, and technically in crocodiles too, although it is a little bit different, we have four-chambered hearts. So we have 2 atria and 2 ventricles, and the ventricle has a complete septum here so that there's complete separation of oxygenated and deoxygenated blood. And this is an incredibly efficient system for sending oxygenated blood where it needs to go in the body. And so think about it as you, I think I got a slide for this. Yeah, here we go. So as you're moving from organisms that are debatably more sedentary upwards to things like mammals and birds that are incredibly active organisms and would, of course, by definition, need a high volume of oxygen to power the muscles. They're going to need a really efficient respiratory system to get that oxygen into their systems. And so a 4-chambered heart is far better at doing that than a 3-chambered or even a 2-chambered heart would be. So there has been an evolutionary influence in the development of cardiac structure. So there's an adaptive advantage to having 2 separate circuits, the pulmonary circuit and the systemic circuit. So to overcome gravity, blood needs to be pumped at a really high pressure, okay? So you can pump, blood out to your heart, out to your lungs, pick up oxygen, and then you can have the ventricle that can pump blood throughout the rest of the body. But if you had to pump it with one structure to your lungs and then out to your body, that's going to put a great stress on your heart. So with a separation, with these 2 circuits, it makes it much more efficient. And you also have the added insult of capillaries and alveoli of the lungs are too thin to be able to withstand the pressure of having a single circuit. So the evolution of 2 separate circuits allow for a high pressure systemic circuit and a low pressure pulmonary circuit. So with 4 chambers, you have no mixing of oxygenated and deoxygenated blood in the single ventricle. You have blood higher in oxygen when it's supplied to the tissues because you lack that mixing, and it supports a much more active lifestyle, allowing for faster production of ATP. So if you don't already know this, let's review the circulatory system as it works in a 4-chambered heart. So blood is going to enter the right atrium as it returns from the body. So it's going to enter through the superior and the inferior, vena cava. It's going to go from the right atrium through the atrioventricular valve into the right ventricle, and it's then going to go through the pulmonary valve out to the pulmonary artery, and this is where it's going to go to the lungs to pick up oxygen. After it's picked up oxygen, it's then going to return to the heart. It's going to return to the left atrium through the pulmonary veins, into the left atrium, through the aortic valve, through the atrioventricular valves into the ventricle, then out the aortic valves, through the aorta, and it's going to go through several different vessels towards your head and then down and through your body. So right atrium, right ventricle, lungs, left atrium, left ventricle, body. And repeat. So this is a diagram from your book that's a little weird to me because this is supposed to be lungs and this is showing muscles. So what it's supposed to be showing you is blood, deoxygenated blood comes to the heart, right atrium, right ventricle, out to the lungs, get some oxygen, back to the heart, left atrium, left ventricle, and then out to the body, and repeat. Now, how does the heart do that? What's causing it to pump? Well, as most of you know, there actually is an electrical current that travels through the heart. So the cells that initiate these contractions are referred to as pacemaker cells, and you have two major ones. It begins with the sinoatrial node, frequently referred to as the SA node, and this is located in the right atrium. So the electrical current is going to be initiated here in the SA node, and when it fires, what it's going to do is cause both of the atria to contract at the same time. So it's a coordinated thing. And so when the atria contract, blood is going to then be forced through those valves into the ventricle. And in this case, again, we're talking about a four-chambered heart, so it's going to go into both sides of the ventricle. When that happens, blood, again, going to go into the ventricle. And after that SA node is fired, there's going to be a short delay. The atrioventricular node, which is in the base right here, it's going to fire, but there's a delay before it fires. And that's in order to allow all of the blood to be pumped from the atria into the ventricle. Once that happens, the atrioventricular node is going to fire, and watch what happens. So it's going to send this electrical current through the ventricular septum right here. It's going to cause this current to contract. The ventricles, remember, these are thick muscles, big thick muscles, forceful muscles. It's going to cause the contraction to begin at the bottom of the ventricles, and the current is then going to work its way up the walls. And so it's going to contract from the bottom to the top of the ventricles, and that's going to make for a really efficient, forceful ejection of the blood out into either the pulmonary circuit or into the aorta to go to the body. And it's going to do that in order to try to make all the blood go out in an efficient manner, and then the ventricles relax. And then it repeats. So SA node, AV node, relax, SA node, AV node. And so this is a diagram that you're probably familiar with that you see when you have an electrocardiogram made of your heart function. This is commonly referred to as an EKG, but in recent years people refer to it as an ECC. It used to be referred as EKG. This comes from the German term for electrocardiography, which is spelled with a K, but of course in our case it's spelled with a C. So you can refer to it as either an EKG or an ECC. And so you can see what's happening in this diagram by referencing what's happening above. So here's the SA node that's firing. Here's the signal spreading across the atria. Oh, I should explain. This is electrical activity in millivolts shown on the y-axis and then time. So you get kind of a little jolt. There's a delay. Then there's a giant signal that goes when the AV node fires, comes down, and then there's a little bit of a signal that goes through when the ventricles relax, and then it repeats. And this is that typical sort of pattern that you see with healthy heart function. So in the cardiac cycle, when you say have someone take your blood pressure, or maybe you go to Walmart and stick your hand in the cuff, there's two numbers that get reported to you. So the contraction phase of the atria and the ventricles is referred to as systole, and the coordinated relaxation of those muscles is referred to as diastole. And so a full cardiac cycle consists of one complete systole and one complete diastole for all of the rooms, if you will, in the heart. So both atria and ventricles together. And so right here in this diagram, I'm just showing you the ventricular diastole and the ventricular systole. But a full cardiac cycle will consist of the contraction and relaxation of both atrial and ventricular, both sides. So when you're measuring blood pressure, it's measured at the peak of ventricular ejection into the aorta. That's called systolic blood pressure. And then just before the ventricular ejection is the diastolic pressure. And so you're seeing that in this diagram here. So the light purple is going to be the aortic pressure. The dark purple is the ventricular pressure. And it's usually measured in the aorta as opposed to in the atria. So you can see the atrial pressure here, but it doesn't change very dramatically. And so so this is diastole, this is systole, and you can see the changes in both the aortic pressures and then in the ventricular pressures throughout the cardiac cycle here, and it's all very quick. So in lab, you're going to get some experience with actually taking blood pressure with a blood pressure cuff and being able to calculate systole and diastole for a person at rest and then a person who's been exercising. And so you should see changes in blood pressure when you measure them. All right, that's all we're going to cover today for the last section of internal transport. On Thursday, what we're going to do is we're going to see how well you understand this material. We're going to do some assessment over all of the internal transport lectures. We're going to have some iClicker quizzes and some activities. And then we're also going to review the results of the exam. So see you Thursday and stay warm.